Hey guys, we are here with chapter 5, which is something that is not review from 101. It's all new stuff, and we're going to start with tissue organization. So we know from our previous lectures, we kind of started with like what is anatomy and physiology, and then we kind of moved on to cells and cell respiration, and that is our first living thing. And then, of course, if we put a bunch of cells together and give them a job, well, then we would have a tissue. So that's where we are here. So let's dive in to our different tissues and how they are organized in the body. First, of course, our objectives. Again, these are also on the syllabus in the D2L for you, but these are the things you should be able to know how to do and explain by the end of our PowerPoint. And you are always welcome to come back to this slide and double check that you feel confident and ready for our quizzes and exams. These are the things you'll be quizzed and tested on. So let's talk about some vocabulary. Obviously, tissues are just a collection of cells and the cell products, things the cells make, that do some kind of specific function together in the body. And histology is the study of tissues. So those interactions between the cells, how they work together to do their function. There are going to be 4 main tissue types. The first is our epithelial tissue. You might think of it like the tissue that is going to cover surfaces of the body, line the organs, the body cavities, and then form glands. We also have connective tissue. Connective tissue is going to fill internal space. It's going to be supporting the other tissues, getting them the things that they need, nutrients, water, oxygen, removing things they don't need, helping them keep shape and stay stuck to the body. So just a support system. They also transport materials and they will store extra energy. We then have muscle tissue. Muscle tissue is specialized to contract, and that really allows for movement, not only of like the limbs, but of things inside the body as well. And we have some categories of muscle tissue. We have skeletal muscle, heart muscle, and then all of the walls of our hollow organs also have muscle tissue. And then we have neural tissue, which is going to carry electrical signals from one part of the body to another part of the body. Let's first start with epithelial tissue and its characteristics. So what we know is that it's going to cover the surface of the body, line body cavities, and form most of the glands. When we look at this tissue, it's going to be a very tightly packed cells, like one right next to the other. So there's very little extracellular space and therefore very little extracellular material. Epithelial tissue is avascular, meaning that it does not have blood vessels. Epithelial cells, they have a free or what we call apical surface. That's the part that's not attached to something else. They have a lateral surface that is attached to other cells. It's like the sides. And then they have a basal surface, which is the part that is attached to a basement membrane or the bottom. There's some special characteristics that happens for our epithelial tissue. We can have a lot of nerve endings or extensive innervation. And that means that in this tissue, there are all of these nerve endings, and that is used to detect changes in the environment. Think of like your skin, right? You can detect heat or cold. You can detect pressure and touch. That's because of all of the nerves. It also has a high regeneration capacity, which means like if it's broken or cut or damaged, it can regenerate. And the basal cells, they're going to go through mitosis to continually replace. So you're building that epithelial tissue like from the bottom up. A couple of structural and functional relationships we should talk about. The apical surface can be modified, and we learned some of this in the last chapter, but it could be smooth, and that is going to reduce friction and help things pass over it without that friction. So an example would be like in your blood vessels. They could have microvilli, which are those little finger-like extensions to increase surface area. And this is going to be very common in cells that either absorb or need to like catch onto, hold on to nutrients. And a great example is in your small intestines. And then cilia can be present, little hair-like structures, sort of doing a sweeping motion that will move material over the cell surface. And a great example is like in your nasal cavity and your trachea. What is the purpose of epithelial tissue? Well, it has many, and you can probably imagine these if you just imagine, your skin, but it's going to be a physical barrier and protection. It will stop dehydration. It's there to protect against abrasions, cuts, and destruction. It is selectively permeable. So some stuff can get through epithelial tissue, while other things, usually large substances, are blocked. It does play a role in secretion, and we do have cells that are specialized to do that. And we call this structure a gland, and we will look much more in depth at glands in the next few slides. And then, of course, detecting sensations, right? We said it has a lot of sensory nerves in order to detect and then produce a response to stimuli. When we look at epithelial tissue, right, I just said skin as an example. We also have to classify the tissue based on the number of cell layers and the shape of the cells. So not all epithelial tissue looks like the skin that you see on your body. We take it under a microscope, look at it, and we know that there are different numbers of layers and different cell shapes. So let's look first at those cell layers. If we say that it is simple epithelium, it just has one layer. That is the most simple, so it's called simple. And that is usually found where we have filtration, absorbing, or secretion. You can have stratified epithelium, which means that you have two or more layers and they're kind of stacked on top of one another. This is going to be found in places where there is a lot of mechanical stress, and it just adds more absorption and more protection to those areas with that stress. And then there's this sort of in-between category that we call pseudostratified. And that means that there is only one layer, but because of how the nuclei are arranged in the cells, it looks like there's more than one layer. It looks like it has two or more. And we'll see coming up sort of where that is found. Then we also have to look at the shape of the cell. So if we have a squamous, then that would be a very thin, flat, almost square-like cell. Cuboidal is a cube. Columnar is tall and thin. And then transitional changes shape depending on the stretch and sort of where that is located on the body. And then to classify these tissues, we put the number of layers and the shape together and make sort of a two-part naming here. So you could have simple squamous or stratified squamous or pseudostratified squamous and so on and so on. So we're going to take a look at some of these, and you also will in your lab class, of course, but we want to dive in here as well. In order to do that, we have this nice little flow chart to help you know where to sort of categorize these different types of epithelial. So we know that we can have simple, which is one layer, or we can have many layers. So that's sort of our first break. When you look at a tissue under the microscope, is it one layer thick or is it many? Break it apart there. If it is one layer, you could have simple squamous, simple cuboidal, simple columnar, or pseudostratified. And that tall column-shaped cells, it can either have the nucleus all aligned, that would be simple, or it's that pseudostratified where it looks like the nuclei are all over the place, so there's more than one layer, but it is just one layer. Okay. And then on the other side of that, does it have more than one layer? Great. You could have stratified squamous, stratified cuboidal, stratified columnar, or that transitional. And if you look at the squamous, there's also a breakdown of, does it have keratin? Are they dead? They're keratinized. Or is it non-keratinized? They are living cells. So we are going to look at all examples here in the next few slides, but this chart I think is useful to organize the different types. After you then see them, you can sort of figure out where they fit, if that makes sense. So let's start with simple squamous. That is just one single layer of flat cells. It's going to let things move across that surface very easily. And that's useful in something like the air sacs in your lungs. They're called alveoli, or something like your blood vessels. Or even when you're talking about serous membranes, we need to be moving air and carbon dioxide. We need to be moving nutrients back and forth. And that simple squamous tissue allows for that to happen easily. And so here's what this looks like. And if you have the Connect software, you can go to Anatomy Physiology Revealed, and you can actually sort of click through some of the pictures there. That's where this comes from. But it's going to give you this little directive too, that it's a single flat layer. You can sort of think of it like floor tiles, right? You can kind of see them all here lined up like that. They're the thinnest possible layer, rapid diffusion and filtration. And so again, in our lungs, in our blood vessels, in serous membranes. Next is your simple cuboidal. So that's our cube-shaped epithelial cells, just a single layer. They're all shaped like little dice next to one another. And they are designed to absorb and secrete, but it's just not as quick as if they were flat. And of course, they are specialized for structures in glands and for doing active transport that requires energy. A great example is lining the kidney tubules, where you're going to be having your blood come through, be filtered out of toxins, retaining water, retaining the blood cells, getting rid of waste. And then also, tiny ducts will have these simple cuboidal epithelium. And so here's what it looks like under a microscope. They do look like little cubes or little dice, one right next to each other, and they are just one layer thick. All right, our columnar epithelial, that's again a single layer of cells. They are kind of tall, taller than wide, so they look like columns. And really, these are great for secreting and absorbing. We can find these like in the small intestines, and other ducts and glands have them as well. And you can see they're very tall, and all the nuclei are in one line. So we know that it is one layer thick. Again, just a little bit more about where we find them. It is going to be in the intestines and stomach. In the intestines, this type of tissue will have microvilli. Your stomach does not. And that microvilli will sort of give it a fuzzy-like appearance. Sometimes it's called a brush border. It's also a place where we have something called goblet cells, and that main job is to secrete a mucus. And of course, that mucus is to help move things through the tissue, as well as can be used as sort of like trapping particles that don't belong. So it's a great use for that. Think like your nasal cavity. Like when you breathe in, it's trapping dust in that mucus so that it doesn't get into your lungs. And again, here's from Anatomy and Physiology Revealed. And of course, feel free to click through it, but this is what it would look like. The tall, sort of thin structure of a column epithelial. We could also have pseudostratified columnar epithelium. It appears like it's multiple layers because the nuclei are sort of all over the place and it's not that perfect column shape. It's a little bit free-formed. You can kind of see on the picture here how some parts are skinnier than other parts and some parts are fatter than the other parts, but they are just in one single layer. It's not actually stratified. That's what pseudo means, false stratified. All the cells will come in contact with that basement bottom membrane. And not all the cells will actually reach to that apical top surface. So that's why we give it this name of pseudostratified. Where are we going to see this? A lot of it will have the apical surface covered in cilia. Again, it will have these goblet cells that can release mucus to trap foreign particles. And we find this in large passageways in your respiratory system. So think like the trachea. And that's really going to have that mucus there and the cilia beating in order to trap particles that we don't want getting into our lungs. Okay, let's talk about this keratinized part of this. Keratinized just means that the uppermost superficial layers are dead. And that is okay because instead of having a nucleus in those cells, we are going to have it filled in with keratin. Keratin, we know, is a type of protein, and it keeps a structure, and it has sort of this fibrous or string-like structure providing support to the cell. What happens is that the cells in the basement, basal, or bottom region, they are going to be alive and they will grow and sort of push up towards the apical surface. As those cells die, that keratin fills them in, and then we have the dead keratinized cells at the surface and the living cells underneath. And this is really what we find in your epidermis of your skin, the outer layer of your skin. And that's when you hear about like sloughing off dead skin. That's that keratinized skin on the top. It's no longer living and you can slough it off. And the under layers are still alive. As they grow, they push up and become those new dead layers. So you're never running out of epidermis, you're just replacing it. Non-keratinized is just cells that are still alive. And this is going to be in moist areas of the body. And really where we have a lot of secretion. So like your salivary glands and in your mouth. They don't have keratin. That is their protective protein. They do have nuclei if you look at them under a microscope. And again, we're going to find these like in your mouth and oral cavity, in your pharynx, esophagus, anus, and vagina. Okay, let's move on to stratified. Stratified meaning that we have two or more layers on top of each other. If it's stratified cuboidal, well, then it's the cube shape, just in more than one layer. This is where we're going to, we're going to find this stratified cuboidal epithelium in different tubes, and then also in any sort of coverings in the body. It is used as protection as well as in secretion. And again, walls of ducts in the exocrine glands. So we're going to talk about glands in just a moment, but exocrine, if you look at that prefix exo, is like to leave, exit, right? So exocrine glands are glands that have ducts that the substance is going to leave the body. And some great examples are sweat glands, parts in the male urethra, and in the ovarian follicles. Then we have stratified columnar epithelial. That's kind of rare. Of course, if there's two or more layers of column epithelium, this is our stratified version. Again, it's going to be sort of a protection and secretion. It really is found in just a few places. You can find it in large ducts in salivary glands. You can find it in the conjunctiva of your eye, and in some parts of the male urethra. Transitional is sort of this mixed thing here, where it can be in a relaxed state where the basal cells are this cube shape, or maybe they're even polyhedral, so many shape. And then the apical has this large round side to it. But this epithelium tissue will change when it becomes stretched. And so then it sort of flattens out on top, and it's what lets there be organs that are sort of movable in shape. So like the bladder stretching to fill, your ureters, anything like that that changes shape will have transitional epithelium. All right, so now that we've talked about the different cells that make it up and how it looks, let's talk about the cells being together, because that's what makes a tissue. So we have to connect the cells, and we do that through what we call junctions. And there's a few different kinds. We've got tight junctions, which, like their name says, binds the two cells together and forms this barrier, where really it's hard to move anything through it. Then there's desmosomes, which are sort of like tying, mechanically binding the cells together. Then there's hemidesmosomes, which also like mechanically bind these things together, but it is the cell to the basement membrane. That's a hemidesmosome, whereas just the desmosomes will be two cells of the tissue together. And then there's something called a gap junction that is going to let things move through these cells. So it allows for intercellular communication. You can think of it sort of like little pores or little holes that allow things to pass in between. Let's talk about glands. A gland is a single cell or a multicellular structure that is going to secrete something. Endocrine, endo, think like inside, endocrine glands secrete hormones, and that just goes into interstitial fluid. And that means that there are no ducts, and whatever is being secreted is just going to go into the bloodstream. Exocrine glands, like we said, exit, to leave. Those are going to produce secretions that are going to go out either onto the epithelial surface or move through a duct. So what kinds of glands do we have? Well, first, let's talk about merocrine glands. Merocrine glands are going to do exocytosis. All right, so if you remember our Golgi apparatus in particular, we said that those make vesicles that then push material out of the membrane. And that is exactly what the merocrine gland is going to do. It's going to use that exocytosis to release a product off of the membrane. A great example, your sweat glands, your tear glands, and salivary glands. We also have something called an apocrine gland. Apocrine, think apo like apex or the top of a cell. Again, we do use our Golgi apparatus to help us do this process, but really we're not just pushing the little vesicle out anymore. We're releasing like the top chunk of cytoplasm off of the cell. And so a great example is mammary glands. This is how they work. When they're releasing their products, it'll kind of build up these little vesicles in the top part. The cell will sort of pinch in, and then it will release that that product. Then we regrow, and we can do it again. We also have something called a holocrine gland. And when you think of the word holocrine, think of like whole cell. And so the cell is actually going to burst and sort of kill the gland cells in order to release its product. We can replace the broken gland cells with stem cells. And so we can keep the process going. But when we have a holocrine gland, we call that product sebum. So sometimes they're called sebaceous glands. And basically, you'll find that in your skin by a hair follicle. It's the oily sebum that is released, and it will actually burst this entire cell in order to release that. And then those lost cells are replaced by stem cells underneath it, growing and pushing up. So again, here is an example showing you the different types of glands and how they get rid of their products. Again, you can have a merocrine gland, which is going to use exocytosis in order to release its product. So that Golgi apparatus is very involved in pushing the product to the membrane and then releasing it out. And that's a lot of like watery type substances, so salivary glands. You can have an apocrine gland, which again is the apex top of the cell, buds off. And that's a lot of proteins, lipids, and steroids happen this way. And in the mammary gland, how we secrete milk is through this process. And then we've got our holocrine glands, where the whole cell is going to burst in order to release the product. And that's like our sebaceous glands in our skin that are releasing like an oily type substance or like a sweat type substance. And again, we replace those with dividing cells underneath. Okay, so that's all that there is on just epithelial tissue. But if you remember, that's just one of 4 kinds of tissue that are the main types that we talk about. So of course, feel free, pause the video, go back through your notes. But let's move on now to our connective tissue. Connective tissue is going to be cells that are separated from each other by a lot of extracellular matrix. We said epithelial tissue has a lot of tightly packed cells. Connective tissue is almost the exact opposite. There's a lot of space. It is still incredibly important because it has many functions. It's going to be involved in enclosing and separating structures. It's going to be connecting tissues to each other, so like ligaments and tendons. It is going to be support and help in movement. Examples, we have our bones and cartilage. It's going to be storage, like fat, adipose tissue, and bones. It's cushioning and insulation, that's fat tissue. It's going to work to transport things around the body, and of course, to protect organs and components of the body. So let's take a look. Again, much more loose, almost like you threw stuff into a drawer, where like epithelial is like you gently placed everything nicely. And really the main parts of it are the cells. Then you've got a lot of protein fiber. And then you've got what we call ground substance that is going to make up our connective tissue. So let's look at the cells that we find. We give them special naming where we use end suffixes to tell us what those cells do. If you see blast or germ, then that is a cell that is from the matrix. That's from that extracellular stuff. If you see cyte, or just cell sometimes, those are cells that work to maintain the connective tissue. If you see clast, that's really there to break down different parts and pieces of that tissue. So for example, some vocab words that you're going to want to know about. A fibroblast, that is blast at the end, right? That's going to produce fibrous filaments and ground substances in the extracellular matrix. So again, if we go back, right, blast forms the matrix. So a fibroblast is making that. Adipocytes, right, that is a lipid or adipose fat storing cell. Macrophages, that is a large white blood cell that is going to destroy pathogens, and it does it by phagocytosis, so like engulfing the invader. We have something called a mast cell. Those are near blood vessels, and they're going to secrete some substances, histamine and heparin, which is going to start this inflammatory response. It's also called paracrine signaling. But like if there is a reason for that mast cell to be inflamed, it's going to secrete those things and tell the body like, hey, something is happening here. Let's go investigate it. Then you've got erythrocytes. That's another name for red blood cells. Leukocyte, that is just a general type of white blood cell. It can do a lot of stuff. It can attack pathogens. It makes antibodies. It's sort of the all-purpose white blood cell. We've got something called a chondrocyte, which is going to be in cartilage, and it has what is called a lacuna. You can kind of think of it like a little hammock, I guess, for the chondrocyte. And then you've got an osteocyte, which is going to be in mature bone, and it also has that lacuna, that little outside structure to it. Okay, so let's break it down now to specific kinds of protein fibers that we find in connective tissues. So we've got elastic fibers. They're going to contain elastin. That's the protein that is there. They are stretchy, but it's always going to go back to its original shape. And we'll find this elastic fibers, or sometimes just called elastin, inside skin and the walls of arteries. Stuff that we want to maybe bend and move some of the time, but we want it to keep its shape. Then we've got collagen fibers. That's the most common fiber in connective tissue. It's very long and straight. It's not branching. It's flexible, but it doesn't really stretch. It actually resists stretching. And we can find this in tendons and ligaments most commonly. We also have reticular fibers, and those are more like a framework structure. And they are going to work especially well in the immune system organs like your lymph nodes and your spleen. And they are able to resist force in many directions because they're more of a latticey type structure. They can kind of be moved in multiple directions and resist that force. Okay, so those are the types of fibers that we find. We also have ground substance, and ground substance is just not cellular. It can be the interstitial fluid. Sometimes there's a more viscous matrix that it's in, and it just kind of depends on the type of tissue that you're looking at. There's something called proteoglycan, which is protein, and that is in cartilage, so it helps to support and keep it lubricated. You can have calcium salts, which give bone strength. So that ground substance is just not cellular. It's the other stuff that is there. And just like with epithelial tissue, the connective tissue has its own flow chart. So we have proper connective tissue, which includes loose connective tissue and dense connective tissue. And then we have different kinds. We have supporting connective tissue like cartilage and its types, and then bone and its types, and then fluid connective tissue like blood and lymph. So we start as this common origin, we call it mesenchyme, and then we want to break it apart. Is it the proper connective tissue? Is it supporting tissue, or is it fluid? And then you can break it down from there. So let's start with the loose connective tissue. We have areolar connective tissue. That's really found throughout the body. It's what's going to let movement happen. It's really found where there's blood vessels and nerves. It's got spaces to have that blood and nerve move through it. It is going to be at the bottom of most epithelial tissue. And a great place that we find areolar connective tissue is the layer of the dermis that we call the papillary layer, which we will go into more depth when we talk just about the integumentary system. We also have adipose tissue or fat. That again is pretty widespread throughout the body. It's going to be an energy storage, a cushion. It insulates, it protects organs. We find it in what we call hypodermis and yellow bone marrow. That's definitely a source in where you can find adipose tissue. We've got reticular connective tissue. This has sort of that sponginess, right? It can be in sort of pulled in every direction. It's going to let there be filtration of fluid. And we find this in a lot of those immune system organs, like the spleen and the lymph nodes, and also in red bone marrow. Okay, let's talk about dense connective tissue. We've got dense regular connective tissue that is very tightly packed. It has these collagen fibers running through it parallelly. It doesn't really have blood vessels going to it, so it will take a very long time to heal if it's damaged. And great examples are in tendons and ligaments. And so if you've ever injured a tendon or a ligament you may even had to have surgery on it, it's a long healing process. And that's because it doesn't have blood vessels going through it to give it the stuff it needs to heal more quickly. Then you've got dense irregular connective tissue. It has collagen fibers, but they go in every which direction. So you can see the difference between regular being nice and straight and orderly and irregular being sort of all over the place. It's going to provide resistance to the stretch in a lot of different directions. So you'll find it in your skin, in the lower layers called the reticular layer of dermis, in the periosteum around bone, that's the tissue that is sort of wrapped around your bones, and the perichondrium around cartilage. Then you've got elastic connective tissue, and you can see that's definitely different. That's able to stretch and then recoil back. And this is great in the walls of large arteries, right? Like your aorta, because you want that to be able to stretch as blood's moving through, but you want it to maintain its shape. You don't want it to enlarge. Okay, supporting connective tissue as a whole, we call it cartilage. And there are three major types. We've first got hyaline cartilage. Hyaline cartilage does not have blood vessels running through it. It's avascular. It is an awesome shock absorber. And we're going to find it in our respiratory tract, in what we call articular surfaces, or where bones meet each other, and the costal region, so between your ribs. We've got fibrocartilage, also avascular, no blood vessels going through it. It's a really good shock absorber, but just not as good as hyaline cartilage. We find this in the discs between your vertebrae and what we call the pubic symphysis. That's the part where your pubic bones are held together. And then we have elastic cartilage, also avascular. That's the flexible, springy cartilage. So you can think of that like the outside of your ear as well as your epiglottis of your larynx. Of course, we have bone. Bone has this mineralized matrix, right? It's got calcium as one of those salts that are there. And it is what makes up the skeleton of the body, most of it. And we've got what we call compact bone that has more matrix than space. And we have calcaneus bone, which has more space than matrix. And we will get into this way more as we talk about the skeletal system. So this is sort of a brief overview. But we call bone cells osteocytes. And because we can have these two types of bones, we have compact bone and spongy bone. And again, we'll spend a lot more time as we talk about the skeleton. Another connective tissue, a fluid connective tissue to be exact, is blood. Blood has this liquid matrix. It is found in your blood vessels, and it's produced in your red bone marrow. So that's where you're making red blood cells. The cells that are a part of this tissue, we have erythrocytes. Those are the red blood cells. Leukocytes, those are the white blood cells that sort of have all purpose. And then platelets, which are just tiny little structures useful for blood clotting. Its main job is to move materials through the body. And what each type of cell has its own thing that it's going to move around. Of course, our erythrocytes, their main job is to move oxygen and carbon dioxide around. Okay, so that is our connective tissue and the different types. Again, we still have two more different types. So again, feel free to pause the video and go through your notes if you need to. But we're going to jump into muscle tissue next. Muscle tissue is specialized to contract, and that is what makes motion possible in the body. There are three types of muscle tissue. The first being skeletal muscle. And skeletal muscle is going to attach to the skeleton and allow for movement. Skeletal muscle is voluntary, which means you have to tell your body to do it. And it's multinucleated cells, so many nuclei in a cell. And we say that it's striated or banded. So when you look at skeletal muscle, you see these sort of ridges in it. Cardiac muscle, that's the muscle that you find in the heart. They're connected to each other by what we call an intercalated disc. And they have these gap junctions so that things can move easily from cell to cell. It is involuntary. You don't have to tell it to work. It is striated, so you will see the banding. And they usually have only one nuclei per cell. So you can kind of see a difference here. This is very, very banded, many nuclei. Not the case for the cardiac muscle. Smooth muscle, that is going to be in the walls of hollow organs, except the heart. The heart has its own kind. And it's also found in the skin and in your eyes. Again, it's involuntary, but it is not striated. You don't see those ridges on it. And it does have just one nucleus per cell. And the cells have more of a spindle shape. So they're kind of pointy at one end, get fat, and then pointy at the other end. And again, we will talk so much more about muscles and muscle tissue when we talk about our muscular system in our lecture later on. Okay, our last type of tissue is our nervous tissue, and that is specialized to have this action potential move across it or an electrical signal. The types of cells we have are neurons. Neurons are nerve cells that actually conduct those action potentials or that electrical signal. And then it has glial cells, which are these usually smaller cells that support the neurons. We're going to find nervous tissue in the brain, spinal cord, and nerves. Okay, so as we talk about tissues, we've got the different types. Now we're going to sort of move into, where are we finding this stuff and what does it do? So we know that we find tissue in our body membranes. It's going to line the body cavities, cover the viscera or the organs, cover the external surface. And we've got 4 types of membranes. We've got mucous membrane, serous membrane, cutaneous membrane, and synovial membranes. So let's dive into what those words mean. We've got mucous membrane, which is going to be lining cavities that are opened to the outside of the body. Great examples are your digestive tract, your respiratory tract, your urinary tract, reproductive tracts. Their main job, of course, is going to have glands in them that secrete mucus. Serous membranes are going to line the basically trunk body cavities or the parts that do not open to the outside of the body. Things like your pleural cavity, pericardial, and peritoneal cavities. They are not going to have glands, but they do secrete serous fluid. Then we've got synovial membranes. Synovial membranes are going to line freely movable joints. So any of your joints that move around, right, like you bend your arm or your knee, you will have synovial membranes. Then you've got the cutaneous membrane, which is your skin. Let's talk a little bit about tissue modification or tissue changes that can happen. So the first is you might have something called a metaplasia. And that is when you have mature epithelium that turns into or changes into a different form. And this could be caused by something in the environment that the body is adapting to. So this could either be good or bad. It doesn't really matter. It's just something is happening and it's changing. A great example is in smokers. We're going to have this metaplastic change in the trachea epithelium. Normally, you've got this pseudostratified, ciliated column epithelial, and then it's going to change and become non-keratinized stratified squamous. And that's just sort of protecting that trachea as you're bringing in more irritants in the smoke that you're bringing into your lungs. This can change back quickly if a person quits smoking. Like it'll just go back to that ciliated columnar epithelial. Okay, and here's what we're talking about. We've got this sort of long cubey shaped cells here. It's got cilia on the top. Okay, we're smoking. We're going to build up instead this stratified squamous. If we stop smoking, we can go back to this type of tissue. It's just there really to protect that pseudostratified tissue. You can also have a dysplasia. This is where we're getting into abnormal tissue development. It could be precancerous. So if you've ever had a test and they say precancerous, they've probably found this dysplasia. And it can revert back to normal. Of course, this is something that you want to continue monitoring by medical professionals. And examples in the cervix, you can have a cervical dysplasia because you've been exposed to HPV. Now, there are vaccines that people get when they're like 12 or 13 to try to prevent this from happening. But in the cervix, you'll have these normal squamous cells. The human papillomavirus is there and infected the cells, and now we've got this change to a precancerous state. And then we can also have something called a neoplasia, which is tissue growth that is out of control. And we can have a tumor. That's when we have this abnormal growth. And it can be benign, so non-cancerous. It has localized growth. It doesn't spread. And that could be something like a skin tag or a mole. Sometimes there will be literature that will even call a wart this. It's just a growth that is not normal, that it keeps growing. Malignant is cancer. And so it will metastasize or spread and invade other tissues. So when you're getting a biopsy done, they're taking a scraping or a sampling of the tissue from this neoplasia, and they're looking at the borders, the edges. Will this keep growing? Will it keep spreading? Then it's malignant. Or does it just have localized growth and not spread? Then it's benign. Sorry, didn't warn you about this icky picture. But we also have necrosis, which is the fancy word for tissue death. And this happens when you have tissue damage that is irreversible. There has been an inflammatory response to a stimuli, and the tissue has been damaged, and it will not go back to how it was. An example is gangrene. We also see this response with frostbite or severe burn damage. We have this tissue death. It will not regenerate. And I hate to leave you on this slide with this icky picture, but here we are. So that is what we need to know about tissues. And I think you will see that a lot of this stuff that we see here is just a little touch or a little taste of what we will dive deeper into as we go through each chapter. So, grand overview about tissues. Now, as we dive into the different body systems, we will look even more in depth. Of course, if you have any questions or concerns, please reach out to me. Otherwise, thank you for watching, and I hope that you have a wonderful day. All right, y'all. See you later.